We are back for topic 2, and now we're talking about how selective forces like natural selection and also random events can change the frequency of different traits within a population of organisms. What you're seeing right here is a very common snail. It's known as a helix snail. You can find them all around the world. This is one particular species that you're seeing, and note that there is great variability in this population. You could go out for a stroll out in the English countryside, for instance. You could walk through fields and pick up the snails and then maybe head over to an adjacent woodland and pick up some more snails. And what you would probably find is that within the fields you see yellow and also greenish colored snails that match the vegetation in that field. In the woodland, you're going to see snails that are, well, darker colored. They're going to be brown and maybe kind of rust color. And they're going to match the leaves that are on the forest floor. They're going to match the soil. And this makes sense because this is what you would expect in terms of camouflage. This variation will allow individuals to do better in slightly different environments, environments that are right next to each other, so micro environments we might call them. And within those environments, they are well camouflaged and they have an advantage. We've talked quite a bit about the development of Darwin's theory. Darwin had a theory that did a good job at explaining what he saw in the natural world, but there were some questions that were unanswered within his book, The Origin of Species, that he talks about. He talks about the idea that I'm missing this piece, I'm missing this piece, etc. One of the big mysteries for Darwin was how inheritance worked. What was the mechanism of inheritance? What was the material of inheritance? And also, was inheritance strong or weak? Now, strong inheritance is inheritance of characteristics that you were born with. So this is the idea that there is genetic material, you're born with it, you pass that genetic material on to your offspring. That genetic material does not change over the course of your lifetime. Weak inheritance is the idea that this genetic material can be changed, and acquired characteristics, characteristics that you develop over your lifetime through use and disuse of structures, can be passed on to the next generation. So that use and disuse somehow changes the genetic information, and those changes are passed on. This is what we would refer to as Lamarckian evolution today, which again is kind of a misnomer because Darwin wasn't sure. In the first few editions of The Origin of Species, he got it right, and he said that inheritance was strong. But later on, he kind of went back to the Lamarckian way of things and suggested that maybe inheritance was weak. In fact, he had a whole theory about this. I'm going to go kind of off on a tangent for a second here. He thought that perhaps all of the different parts of the body, the different structures, produced little packets of information. He called them gemmules, and they were released into the bloodstream, and they got together to help form the sperm or egg, and they were passed on. And if an organ changed, it could change the contribution of genetic material to these little packets. It's a pretty interesting theory, but of course, it's completely wrong. So biologists tend to ignore some of the additions he made to later editions of The Origin of Species. Now, like begets like, to kind of quote the Bible and quote the kind of poetic wording that Darwin would use. And what I mean by that is, organisms give birth to things that look like them. Your parents look somewhat like you, but they don't look exactly like you. And that was the real puzzle here, because back in Darwin's day, the main theory of inheritance was a blending hypothesis, where basically parents will donate genetic material and that gets blended and we get our offspring. But we know that that's not quite right. You are not the average of your parents. You are not the average of their appearance. And some traits are very distinctive. They're granular, I guess you could say. They're all or nothing. Like for eye color, there are only certain colors that are possible. For, let's say, well, he wouldn't really have been dealing with this, but blood typing. There are certain blood types that you can have. It's not just purely a mix of everything, and we'll come back to that as well. Now, there was someone out there that had some of the answers, and you've probably heard this name before, Gregor Mendel. Mendel was around at the same time as Darwin, and he had come up with what we now call Mendelian genetics, but no one appreciated what Mendel had. And Mendel went largely unnoticed and forgotten, and his work was discovered quite a bit later. There is a story that Mendel's paper was sitting on Darwin's desk and he never got around to read it. And I hate to burst any bubbles, but that's probably entirely an urban myth, so to speak. What we're talking about today, population genetics, was something that Darwin had no idea about. No one did. This was a field that didn't really open up until well into the 1900s. I think it's useful now that we go back and talk about Mendel very briefly. And I'm hoping this is kind of a refresher, but if you need more of a refresher, I do have some other videos on this you can watch. Mendel realized, and again, he lived at the same time as Darwin, he realized that there were these discrete packages of information that were passed from parent to offspring. He didn't know anything about genes, but these discrete packets he was talking about, we know now that they are genes. He did a classic experiment, several classic experiments with pea plants, and you're seeing one of them here on the right. He took purple flowers, and these were true breeding purple flowers. What that means is he had bred them over and over and over, crossing them with themselves for generations, and they always came out purple. And he had some true breeding white flowers, and he crossed them together. And in the second generation, this so-called F1 generation, he ended up with all purple flowers. What he proposed was that the purple flowers had these genes, again, he wouldn't have used that term, that could override the effects of the genes in the white flower. When he took that F1 generation of purple plants and mated them amongst themselves, he got mostly purple plants, mostly purple flowered plants, and a few white flowered plants. And he saw this in a 3-to-1 ratio. And he proposed that, again, there was this dominant factor that coded for the purple color, and it masked the effects of the factor that coded for the white color. And the plants in the second generation, they had that dominant factor. They also were carriers for what he called the recessive factor. So looking on the left here now, let's take a look at how this actually works. That purple color in the flowers is the result of a purple pigment. What happens in the flower is there's a colorless compound that is converted into a purple pigment by an enzyme. And there's a gene that codes for that enzyme. If the gene is working correctly, if it's all there, it's not broken, then it's going to code for the functional enzyme, and we're going to get purple pigment and purple color. If that gene is broken, if it doesn't work at all, we're going to get enzyme that doesn't work. We don't produce any purple pigment, we don't have any pigment, and the flowers look white. So we call that gene that's fully functional the dominant form, and we use an uppercase letter, so in this case P, to signify that, and we call the broken form of the same gene the recessive form, and we use a lowercase p. You don't need much in the way of enzymes, you just need a tiny pinch. So if you just have one working gene, you're okay, that's enough. Pea plants and most eukaryotic organisms, as we'll see, almost all of them are diploid. And what that means is they have two copies of genes. So for instance, in us, you have two sets of information in all of your cells, except for sperm cells and egg cells. You have one set of information that came from your father's sperm, one set of information that came from your mother's egg. Now let's say you've got a gene that was donated to you by your father and it's broken. You might be okay because the gene that you got, the copy that you got from your mother, if it's okay, it's enough to compensate for it. And in that case, the one you got from your mother would be the dominant. The one you got from your father that was broken, that would be the recessive. We talk about these alternate versions of a particular gene as alleles. So we have the dominant allele. In most cases, that's an allele that's working just fine. That's the form of the gene that is normal, quote unquote, I guess you could say. It's also referred to as the wild type form because it's usually the one that's most common out there in the wild, in a wild population. And then we have the recessive form, which usually is broken. It's not working properly. So looking again at that experiment by Mendel, you can see exactly how this works. If we have at least one dominant allele in the pair of alleles for that gene, then we're going to get this dominant trait. One more thing I should mention before we go on, I'm going to throw a couple more terms at you here. Phenotype, that refers to what the outward appearance is of the organism, or sometimes the behavior of the organism. So is it purple or is it white? We're talking about phenotype there. Genotype refers to what alleles we have. Do we have big P, big P? Do we have little p, or do we have big P, little p? I'll come back to this and again. I know I'm kind of overwhelming you with some terms here, but if we have two alleles that are the same, so big P, or little p, that is referred to as a homozygous condition. Homo means the same, zygous refers to the zygote, so the sperm and the egg had the same allele, they came together, and we just see that one type of allele. Or we could have heterozygous. Hetero means different, so that's where you had the big P and the little p, and again it's because different alleles were contributed by the sperm and by the egg. We can use what's called a Punnett square to figure out the outcome of a cross. And what I mean by a cross is a mating. So when we take two individuals, pick them and mate them together, that is referred to as a cross. What you're seeing here is a Punnett square for that particular gene we were just talking about, the one that codes for the purple color. Let's say we take two heterozygous individuals. Again, that means that they have the big P and the little p. They have the recessive form of the allele and the dominant form of the allele. If we take those two individuals and we cross them, plants, just like us, they produce sperm and egg. The sperm are in the pollen grains. The sperm are going to be both big P and little p. So the plants themselves are diploid. That means they have two sets of information, but they're going to undergo a type of cell division known as meiosis. And that creates haploid gametes. Haploid gametes only have one allele. So again, the only choices for the sperm is they can be little p or they can be big P. They can't be both. And the same for the eggs that are found in the flower. We have big P and little p, again, because we're starting out with a diploid heterozygous plant. When we cross them together, all we have to do is take those alleles and combine them to get our diploids. You can see that we have one plant that is big P, big P. We would say that is homozygous dominant. We have one plant that is little p, little p. We would say that that's homozygous recessive. And then we have two plants that are heterozygous. Heterozygous means they're big P, little p. They're both recessive and dominant, but the dominant allele overrides the effects of the recessive. And what we're going to get are three plants that are purple and one plant that is white. Now that's 3-to-1 ratio, just applies to the outcome for the entire mating that you're going to do. So you could cross a whole bunch of flowers and maybe you'll get, you might get a few thousand flowers, but you're going to see this distribution of purple and white in this 3-to-1 ratio. All right, so now that we've done this quick little refresher on Mendelian genetics, let's dive into population genetics. And a couple more definitions for you here. So what is a population? A population is members of a species within a given area. Humans are a species, and we could take a look at this satellite image of the Earth at night, of North America at night, that you see on the right. We see all those areas that are lit up. And this is actually showing light pollution produced by cities at night. Each one of those really bright lights is a major city. I've put an arrow on one, that's Vancouver in British Columbia, Canada, and you can see that city down below. We could set that as our area, and then we could say, okay, let's look at all the raccoons in that area. Let's look at all the red squirrels in that area. Or we could say, let's look at all the humans in that area. So a population is a collection of individuals of one species living in a defined area. And as a research biologist, you could define that area however you want. What's a species? Well, a species is the largest population on the planet that can interbreed and produce viable offspring. We'll come back to that later. What do I mean by that? Well, humans, if we look at the entire planet and consider the entire human population of the planet, that would collectively be all the members of the human species. A species, by the most common definition, can't interbreed with another population of a different species. So for instance, humans and orangutans cannot interbreed and produce offspring. That's how we know they're two different species. Just a few more definitions, bear with me here before we really get going. Allele, we've seen that already. So an allele is an alternate form of a gene. For our purposes, to keep things simple, we're only going to deal with two alleles for any gene. We'll have one that's fully functional, that's the dominant form, and we'll have one that's completely non-functional, and that will be the recessive form. But do realize that we can have other types of alleles for a gene. We could have, for instance, an allele where the gene is hyperactive. It's actually too active. It works too well. We could have a version of a gene where there's some activity, there's limited activity. We could have a version of a gene where the enzyme, for instance, that it codes for does something a bit different than normal. But again, we're going to keep things simple, we're going to just assume we only have the two possible alleles for any gene. Gene pool. Now the gene pool, you've probably heard that term before, refers to the entire set of alleles found in a population. It's all the information, all the available information that's out there in a population. Allele frequency refers to how common an allele is in the population. As you might imagine, for our examples, generally we would expect to see the dominant form more frequent because usually the dominant form is the form of the gene that works best. Finally, microevolution. What is microevolution? It actually has a pretty simple definition. It's changes in allele frequency over time. So the allele frequencies can change from generation to generation. That's going to affect the phenotype, which is the outward appearance of the organism. In fact, it kind of works the other way around. So natural selection, for instance, might select a certain phenotype, and that is going to make the allele that codes for that phenotype more common generation after generation. But microevolution, we're not talking about the appearance of new species. We'll get to that yet a little bit later. We're not talking about macroevolution, which is major changes, the development of brand new structures, that kind of stuff. Microevolution is something much smaller. It's just changes in allele frequencies within a population over time. And now it's time to introduce Hardy and Weinberg. Hardy on the left was a mathematician. Weinberg on the right was a doctor, a gynecologist actually, and the two of them independently came up with a theorem that allowed them to predict the frequency of alleles in a hypothetical population. Now, the first thing they both noted was that breeding alone will not change the frequency of alleles in a population. Think of it like a deck of cards. You can take cards, you can shuffle them, you can deal them, you can take them back, you can shuffle them again. None of that is going to change the frequency of the cards in that deck. So you're not going to change the frequency of queens, for instance, in your deck of 52 cards. The gene pool and the frequency of alleles in a population has to change due to other forces. So their theorem only applies to a hypothetical population where really nothing is changing. They were looking at a hypothetical population of diploid organisms that mated randomly and didn't emigrate or immigrate to other areas. There was no natural selection. There were no outside forces acting on this hypothetical population. The Hardy-Weinberg theorem is also referred to as the Hardy-Weinberg equilibrium for that reason. Things are not changing, and we're dealing again with a hypothetical, non-evolving population. Let's do a little bit of math to demonstrate this theorem. Don't worry, it's going to be basic math. It's not going to be painful. Trust me, I promise. Here we have a hypothetical population of flowers. These plants are diploid. Remember, that means each cell in these plants contains two alleles, and they come in two colors. So we have red flowers and we have white flowers. There's lots of red flowers, there's only a few white flowers. The pigment is determined by the effect of one gene, and we're going to use the letter R to represent that. The big R represents a gene that codes for a fully functional enzyme that can make the red pigment, and the little r represents a broken version of that gene. It can't produce any pigment. And of course, the big R, the functional allele, is dominant to the recessive. First of all, let's calculate the phenotypic ratio. Now in this chart to the left, they've broken it down by genotype. So we have red individuals that are homozygous dominant, big R, big R. We have red individuals that are heterozygous, big R, little r. We have 320 of the homozygous dominant, 160 of the heterozygous. We add those together, we get 480. We divide by the total number of plants, which is 500, and we're going to get 0.96. That is a frequency. We could also represent that as a percent. It would be 96%. For the white flowers, we have 20 of those individuals out of a total population of 500. So that's 0.04. or 4%. 96 and 4, that adds up to 100%, so that's a good way of checking to make sure that we did things right. Now let's look at the genotype frequencies. So for the homozygous dominant, we have 320 of those individuals. As you can see highlighted in yellow there, if we take 320 and divide it by 500, we get 0.64. That is our frequency. We have 160 of the heterozygous individuals. Again, we can divide that by 500. So 160 divided by 500, we get 0.32. And then we have 20 of the homozygous recessive individuals. We divide that by 500 and we get 0.04. 0.64 plus 0.32 plus 0.04, that adds up to 1, so it works. Again, we could think of this as 64%, 32%, and 4% if you'd like. But with frequencies, we generally use decimals, and they have to add up to 1. Now, let's see if we can figure out the frequency of the two alleles, the dominant and the recessive, in the entire gene pool. We have to consider how many alleles of each type each of the three genotypes contributes. First of all, we have 500 individuals. They are all diploid, meaning they have two alleles. So we have 1,000 alleles in the entire gene pool. Let's start with the dominant allele, the uppercase R. We have 320 individuals that are homozygous dominant. They're each contributing 2 of those dominant alleles, so that's 640. But then remember, we have the heterozygous individuals. There's 160 of them. Each of those individuals is contributing 1 dominant allele. So we take the 640 and we add the 160, we get 800. So 800 dominant alleles out of the 1,000 total alleles in the population, we get a value of 0.8. Now we could at this point just subtract 0.8 from 1 and we'd get 0.2 for our recessive allele, but let's do the math on the recessive allele as well. So for the recessive alleles, we have 20 individuals that are homozygous recessive. Because they're diploid, they are going to contribute 40 recessive alleles, little r's. And then once again, we have our heterozygous individuals. There's 160 of them. They each have 1 recessive allele, we add the 160 with the 40, we get 200 out of 1,000, and yep, 0.8 plus 0.2, that adds up to 1. This time, let's work backwards. Imagine that I know the allele frequencies for a population. So I know the frequency of alleles in the gene pool. I can use that information to figure out the genotypic frequencies and the phenotypic frequencies for that population. To do that, we're going to use a Punnett square, but it's a bit different than how you've normally used a Punnett square. Normally, when you're using a Punnett square, you have two specific individuals that you want to mate. You know their genotype, and what you're going to do is figure out what alleles they can contribute in their sperm or their egg. You're going to cross that in the Punnett square and figure out the possible offspring that they could produce. Well, here we're using the Punnett square as a probability table. Imagine that I stepped out into our hypothetical field of flowers with a blindfold on, and I bent down and I picked up a flower. I can't see it, I don't know what color it is, I don't know anything about it, I don't know its genotype. And I take a few steps and I bend down and I pick up another flower. Now, the flowers that I've picked up, for each of them, there's an 80% chance that they could be a plant that could contribute a dominant allele, and there's a 20% chance that they could contribute a recessive allele. So you can see I've added these values to the axes on our Punnett square. So we have 0.8 as a frequency for the dominant allele, and as we'll see in just a moment when I introduce the Hardy-Weinberg equation, we will assign the frequency of the dominant allele as P in that equation. There's a value of 0.2 for the frequency of the recessive allele. That is given the symbol Q, again, in the Hardy-Weinberg equation. Now we cross these in our Punnett square just like we would do normally. So if we cross one dominant allele from the sperm, one dominant allele from the ova, just the egg, bring those together. Again, we have our 0.64 value. And in the equation, that's going to be p squared, p times p. For our homozygous recessive, we're crossing a value of 0.2 with a value of 0.2, and we're getting a value of 0.04, and that's q squared in our equation, again, as we'll see in just a second. Now there's two ways on this chart to get our heterozygous condition. So we have 0.2 times 0.8, we're doing that twice, and we're adding that together. So our value for the heterozygous individuals, that's 2pq. So it's p times q times 2. Now once again, all of this has to add up to 1, and it does. We can also calculate the phenotypic ratio quite easily. All we have to do is add up the different cells from the Punnett square. So all the cells that give us a white flower and all the cells, all the boxes in the Punnett square that give us a red flower. So for white, it's 0.04. So looking at our axes there, 0.2 times 0.2. And then for the red, of course, it's everything else. So it's 0.64 plus 2 times 0.16. And you might notice that the simplest way to do this, if you just want phenotypic ratios, is to take the allele frequency values for the recessive allele, multiply them together, so square them, so 0.2 times 0.2, that gives you 0.04. subtract that from 1, and that will give you your 0.96. You don't even have to worry about the fact that you're dealing with homozygous dominant and heterozygous. You can just subtract that 0.04 from 1. Taking what we just saw from our Punnett square, which again was actually a probability table, let's figure out the Hardy-Weinberg equation. Before we do, first of all, just note that the allele frequencies all have to add up to 1. The genotype or genotypic frequencies have to all add up to 1. The phenotype or phenotypic frequencies all have to add up to 1. So looking at the allele frequencies, we have P, which represents the frequency of the dominant allele in the gene pool, plus Q, and Q represents the frequency of the recessive allele in the gene pool. P plus Q have to add up to 1. So if we know P but we don't know Q, not a problem. We take P and we subtract it from 1, and that will give us Q. If we know Q but we don't know P, also not a problem. We can take Q, subtract it from 1, and that will give us P. All right, here's the actual equation. And again, it's really straightforward if you just think about it. The genotypic frequency for homozygous dominant individuals is P squared. It's just P multiplied by itself. The genotypic frequency for homozygous recessive is Q squared. It's just Q multiplied by itself. And for the heterozygous individuals, again on the Punnett square, we had 2 ways of arriving at a heterozygous individual. So it's 2 times PQ. So again, don't get too concerned about the math here. It is actually a really, really simple equation. Remember, the Hardy-Weinberg equation or theorem applies to a hypothetical population, and there are a number of criteria that have to be met. We're talking about a population of diploid organisms, and we're talking about a situation where there's only 2 alleles. So we're just talking about a single gene. There's 2 alleles and one is completely dominant to the other. There's a bunch more criteria. The population has to be very large. In fact, infinite. The population has to be isolated. And what I mean by that is there's no gene flow, as we'll talk about. There's no movement of alleles between your population and another population. So alleles aren't leaving and they're not coming in. Mutations can't occur. Mutations create new alleles. Also, the mating is random. There's no sexual selection going on, and there's no natural selection. This doesn't describe any natural wild population. It can be useful with actual populations, like human populations, for instance. It's used in medical research to calculate allele frequencies, but here we're talking about using it over short periods of time and not from generation to generation. The main function of the Hardy-Weinberg equation or theorem is to act as kind of a baseline. It's to act as almost like a negative control. You go out and you look at a real population and you'll find it doesn't match the Hardy-Weinberg equilibrium, and we can use that fact to identify changes. And these changes are the result of the forces we just talked about. Sexual selection, natural selection, the sampling error that comes with small populations, etc. Now, before we go on and look at those exceptions and talk about how they bring about change in real populations, I thought I'd just do one more quick example of the applications of the Hardy-Weinberg equation. Now, what I've got here are some hypothetical critters, some fictional critters known as shmoon, or if we're talking about one, or shmoo. This came from a very popular cartoon. The very first cartoon was actually a political cartoon back in the 1930s and 40s. And this was right around the time that population genetics was really taking off. And if you look at old textbooks, you'll see lots of examples that students can work through that use these shmoon. You don't see them very much anymore, so I'm trying to bring them back from extinction here, and they're pretty easy to manipulate in Photoshop. Anyway, imagine that you're walking through the woods and you come upon this valley and you find this isolated population of shmoon. We have wild type. Wild type refers to the norm, the phenotype in this case that is most common in the wild. But then we also have a small number of blue spotted shmoon. The wild type is going to be homozygous dominant or heterozygous. So again, we've got one gene that's totally dominant to a recessive gene, and the blue spotted shmoon are the product of a homozygous recessive condition. And I've given you the genotypic frequencies here. And again, they all have to add up to 1. So 0.81 plus 0.18 plus 0.01, that adds up to 1. And the phenotypic ratio also, of course, adds up to 1. So you can see the blue spotted shmoon are very rare, 1% of the population. Now I've given you everything in this example. And of course, if it was a test question, I wouldn't do that. I would have you use these two equations to figure out the missing values. So you have, again, your Hardy-Weinberg equation. You have p squared plus 2pq plus q squared equals 1. And we also have our p plus q equals 1 that describes the alleles in the gene pool. You can probably see that I only would have to give you a value for the recessive allele or give you a value for the recessive genotype, so give you the genotypic frequency of the homozygous recessive, and from that you could figure out everything else. With the dominant phenotype, there's a bit of a wild card there because you don't know how many homozygous dominant you have and how many heterozygotes you have. But if you have the homozygous recessive phenotype, you can figure out everything else. So let's say that I gave you that value. It's 0.01 for the homozygous recessive genotype. From that, you know that that is q squared. So how do you figure out q? You just take the square root of that. And then you have 0.1 for your recessive allele frequency. And then from that, of course, you know that p is going to be 0.9. You have those two values. You can figure out everything that is missing. Remember, when you're using these equations, they have to add up to 1. So that's how you would check to make sure that you've done everything correctly. To stress just one last time, the Hardy-Weinberg theorem applies to hypothetical populations, not really natural populations. It might be useful if you're looking at a natural population and you just want to figure out the allele frequencies at this moment. So just looking at a natural population, kind of a quick snapshot in time. But I can't use it to predict what the allele frequencies are going to be 20, 30 years from now, because presumably there's going to be other forces at play that are changing the frequency of those alleles. And that's what we're going to look at right now. The first thing that will change allele frequencies over time from generation to generation is just random chance. And this is something known as genetic drift. Genetic drift is most prominent in small populations because it's the result of sampling error. Gene flow, just what it sounds like. This is the flow of genes, so the flow of alleles out of a population or into a population. So we're talking about emigration with an E and immigration with an I. Mutations. Again, mutations change alleles and introduce new alleles. We might have 2 alleles, and now with a mutation we have 3. Non-random mating. If mates are not made equally, I guess you could say, if some potential mates are more attractive, they're sexier, they're more likely to get selected for mating, then the alleles they have are going to increase in frequency in the next generation. And of course, the big one, natural selection. Natural selection is going to change allele frequencies over time. Here is a very simple example of genetic drift. Again, using our hypothetical population of red and white flowers. So we can see that we have 10 individuals in generation 1 on the left. We can figure out the allele frequencies by just looking at our different genotypes here. And if you're not sure how we arrived at those numbers at the bottom, maybe pause the video for a second and figure it out. Okay, so how do we do this? How do we figure out the frequencies of the dominant allele and the recessive allele? It's pretty straightforward. We have 10 individuals. They are diploid. That means we have 20 alleles in our entire gene pool. It's a tiny gene pool. If we were to look at these genotypes and just actually add up the big R's, the dominant alleles, we would find that there are 14 dominant alleles out of 20. So that's our 0.7. Likewise, we could add up all of the recessive alleles, the little r's, and we have 6 out of 20. And that's our 0.3. Now, in this example here, only half of the plants are going to get to mate and leave behind offspring. And notice that the 5 that are going to leave behind offspring, they've been highlighted with a white box there, don't represent the entire population. And this is just by chance. This is sampling error. Sampling error is where you have a small collection of individuals, and just due to chance, quite often that small segment of the entire population isn't representative of the entire population. Just the fact that we have included 3 heterozygotes and the one and only homozygous recessive, again just by chance, that is going to lead to an increase in the frequency of the recessive allele in the next generation, as you can see. Here we have 2 examples of extreme sampling error. Sampling error is something that's very important in statistics if you've taken any statistics. To give you an example before I get to these, let's say you were to flip a quarter. Now, if you flipped the quarter let's say 4 times, and it came up heads every time, that's not too crazy because this is a very small sampling. If you flipped a quarter 10,000 times and it came up heads every time, that would be very strange. So sampling error very commonly brings about frequencies that you would not expect for a larger sample. The bottleneck effect is where we have a population that experiences a catastrophe of some sort. So we have a disease coming through, we have a forest fire coming through perhaps, and killing off most of the population. So maybe we have cute little bunny rabbits and a forest fire comes through and 95% of them are wiped out. It wouldn't be too crazy to expect those 5%, the remaining rabbits, to not necessarily represent all the diversity we saw in the original population. In the example there at the top left, we're seeing beads that represent different alleles. We've got a bottlenecking event that leaves behind a small number of individuals. You can see that the yellow allele, which was pretty rare, is completely gone now. And you can see that the blue allele is overrepresented compared to the original starting population. So disasters can lead to very, very rapid genetic drift, the random change of allele frequencies. The founder effect is very similar. So imagine we have an ancestral population of moths or butterflies in this case, and some of them find their way to a brand new island, an island that has recently, by recently I mean, maybe a billion years ago, popped out of the ocean due to volcanic activity. A small number of these moths get blown off the mainland and they find their way to this new island. That small number of individuals, again, due to sampling error, won't necessarily be representative of what we started with. So going back to our example with the red and white flowers, founder's effect, this is where we're talking about individuals migrating to a new area, a brand new environment. Happens quite often with plants. They release seeds into the air and those seeds can be blown quite far and find their way to a new island or perhaps an area that was just devastated recently by a forest fire, something like that. So in this example, we just happen to have only homozygous dominant individuals finding this new environment. And just with that one event in this new environment, we don't have that recessive allele. We would say that the dominant allele has become fixed in this third generation. So in the third generation, there's only the dominant allele. There's no recessive allele. The dominant allele is fixed. It is the only allele present. Using our red and white flowers again, but this time looking at the bottleneck effect, we have a major catastrophe that just happens to wipe out the heterozygous and the homozygous recessive individuals. Again, just due to chance, we're dealing with a very small population, so, this is something that could happen and wouldn't be surprising. We once again are ending up with the same situation where the organisms haven't moved this time. That's the big difference between the founder effect and the bottleneck effect. We don't have individuals moving to a new environment. Instead, a catastrophe has hit that area and the remaining individuals give rise to the next generation. And once again, that dominant allele has become fixed. The frequency is not going to change unless we get, other things happening. Let's have a look at a very tragic catastrophe, a man-made catastrophe. It's thought that when Europeans first came to North America, there was about 80 million bison. During the 1800s, that number was reduced to about 1,200 or less. What you're seeing in this photograph is a mountain of bison skulls. There were so many bones that they would mine them for phosphorus to make gunpowder and other things. People would shoot at bison from moving trains. It was just sport and waste. If we have a look at the map here of their range, it's astounding how quickly their numbers were reduced. In brown, you can see the original extent of the bison herds. They covered much of North America. In 1870, it was reduced to what you see in beige, and only 19 years later, in 1889, it was reduced to those small red patches. The numbers that you see in bold are the number of individuals in those herds. So we had some herds that were 20, 25, 10 individuals. A large herd survived up in northern Alberta in what is now Wood Buffalo National Park. As you might imagine, a great deal of genetic diversity was lost forever. So the thousands of genes that are found in a bison, those genes have different alleles, and many of those alleles are gone for good. Conservation biologists have gone to great lengths to try to conserve what's left through very careful breeding programs. And we'll talk about that more when we talk about ecology later in the course. Gene flow. As mentioned earlier, it's pretty much what it sounds like. It's the flow of genes, the flow of information between populations. We might have new alleles coming into a population through immigration, or we might have alleles leaving through emigration. In many natural populations, there are barriers to gene flow. We can have two populations that are physically isolated by an ocean, by a mountain range, and there's no gene flow between them. If we look at human history, we have had human populations that have been isolated from each other, sometimes by geography, so physical barriers, but also quite often by cultural barriers. And for that reason, we do see distinct races and differences. Now, fortunately, there is a lot of mixture. A lot of gene flow is going on in the global human population right now. And we might talk about this a bit later, but having a diversity of alleles in a population, having a diversity of alleles in an individual is a good thing. It creates something called hybrid vigor. If you're fighting a new disease, or if you're exposed to a new environment or something like that, and you have diverse alleles within your body, or if we're talking about a population within the population, those populations, those individuals tend to do better. Gene mutation. This is a big one here. Again, we're talking about how allele frequencies can change within a population over time. Gene mutations create new versions of genes. They create new alleles. And without mutation, evolution wouldn't happen. These new alleles can be selected for or against by natural selection. And again, that brings about evolution over time. The vast majority of gene mutations are bad, but very rarely they might be helpful. So in this diagram here, you're seeing the normal gene, and it codes for a protein. Again, proteins do all the work in the cell, in your body. They bring about the phenotype. A mutated gene can result in no protein being produced. That's called a null mutation, N-U-L-L, meaning nothing. Nothing is created by that gene now. Or we can get an abnormal protein. That protein might not work very well, but it also maybe might be able to do something new. Recall that random mating has no effect on allele frequencies. Non-random mating, or sexual selection, definitely does. If there are certain alleles that make a particular individual more attractive to the opposite sex, that individual is going to be more likely to mate. They're more likely to leave behind offspring, and probably more offspring, and that's going to increase the frequency of those alleles in the next generation. Alleles that hurt your chance of mating are going to be selected against, and they might disappear over time. Also remember that sexual selection leads to sexual dimorphism quite often, and that means that the males and females look different, as you can see with this example here, black widow spiders. Finally, we have natural selection. Generally the most important of these factors when it comes to evolution. Individuals are favored that are well suited to their environment, that are best able at competing with other species and other members of their own species so they can gather resources, and individuals that are generally good at avoiding predation and disease and other things that might limit their survival. Again, it comes down to mating. They do have to mate and pass on their alleles. Not all individuals survive to mate. The ones that have these advantages are far more likely to do so. Those that don't, of course, their alleles are lost. Let's look at a silly example. Let's say you're having some people over, bit of a party, you're putting out snacks, and you put out a big bowl of jelly beans. Very important research tells us that red jelly beans are very popular. Most people like red jelly beans. Black jelly beans, not so much. A lot of people do not like black licorice. So you could do a little experiment and you could figure out the phenotypic frequency for these different colors before your party, and then look at what's left in the bowl and do the same thing after the party. You would find, probably, if the research is correct, that the phenotypic frequency of the red jelly beans has dropped a lot, and you would find that the black jelly beans, not so much, unless you invited me to your party because I quite like black licorice. Anyway, though, this is a pretty good model for predation. Some phenotypes are more susceptible to predation. Now, jelly beans don't have genetic material and they don't reproduce, so we can't take it any further than that. But let's take a look at a more meaningful example. Insects. So we have some beetles here on the left, and in any population, we're going to see variation. Remember, organisms are born with variation that is inheritable. Insects that stand out, that don't match their surroundings, are going to be more easily spotted by birds. They're more likely to get eaten. It's possible that over time we may see some climate change going on. We may see a change in the microenvironment that these guys are living in, and maybe the color that they need to match, the color of the substrate, might change as well. Either way, the insects that are better camouflaged are less likely to get eaten. And we would expect the alleles that code for those helpful colors would become more frequent from generation to generation. Acting over a long enough time period, natural selection can be a directional force. So it can start to shift a phenotype very slowly to something a bit different. In this example here, in the top graph, you're seeing a population of mice. And there's quite a diversity of fur color in these mice. That blue line, that bell curve, is showing us the distribution of colors in these mice. So most of the mice are in the middle there, that kind of medium brown. But we have a small number of variants that are very light and a small number of variants that are very dark. Let's take a look at scenario A here down the bottom. Let's say we get a change in the environment. We get a slight, slow climatic change to the environment. Maybe the soil changes. We have a change in the color of the soil. It becomes a bit darker. We get selective forces pushing that population towards the darker colors. That would be directional selection. Let's skip B for a second and look at C. Let's say that that environment changes and perhaps we've gone from a heavily wooded area to a more open area, and perhaps these mice now are very susceptible to predation from birds of prey, hawks and eagles and owls and so on. And let's say that medium brown is the best match to the surroundings. We will have very strong selection against the outliers. If you're really light or really dark, you're going to stand out. So it's a stabilizing selection, and it's actually reducing the variability in this one trait. Let's go to B. So in B, we have diversifying selection. So perhaps these mice have started to colonize different microenvironments. Within any environment, there's going to be areas that are slightly different, and there's going to be different camouflages that work well. So here we have selection for the light color in one of these microenvironments and the dark color in a microenvironment. Maybe they're leaving the original area and venturing out into new environments, for instance. And we have selection against the middle. You can probably see where this is going. Over time, those two subpopulations might become quite different. They might become entirely separate populations with limited gene flow between them. That's what's happened here. This is an island in Hawaii, and we're looking at two populations of mice. One shown in yellow, one shown in red. They started out as the same population, but they went their own way. We have some mice that have migrated towards the coast, some that have migrated to the upland mountain areas. And we have quite a lot of geography between them now. We have a mountain range in the middle that is physically isolating them. They haven't come into contact with each other for a long time, and there hasn't been any gene flow. Gene flow tends to average out any changes. If a new allele pops up in one population, it can be shared with the other population and vice versa, but they have been separated. Their DNA hasn't changed per se, and in fact, if we were just looking at DNA sequences alone, we might say they're still the same species. But the arrangement of the DNA has changed. This is not uncommon. DNA can be swapped amongst chromosomes. Big chunks of chromosomes can translocate to a new chromosome. So if we look at the chromosomes, that's what we're seeing here. If we look at the top for the yellow mouse, we have chromosome 1, and it's the same. It hasn't changed in our other mouse. The next chromosome there is 2.4. Now that used to be chromosome 2, but chromosome 4, which you can see is gone in the top mouse, part of it was transferred to chromosome 2 to give us 2.4. And the other part was transferred to chromosome 3 along with part of 1 to give us chromosome 3.14, etc. All the same information is there, but it's being arranged differently. And what that means is now, even if these mice were brought back together, if you caught one from one side of the island and one from the other side, and you threw them into a cage together, they certainly might try to mate, but they won't have any offspring. And the reason for that is the sperm and the egg that they make now are not compatible because of the rearrangement of the bits and pieces of the chromosomes. And at this point, we could say that they are different species. But that's something we're going to talk about in our next topic. Just a few concepts here. Evolution occurs at the level of the population. And remember that microevolution simply refers to changes in allele frequencies within a population over time. The phenotype of an organism is its outward appearance, maybe its behavior in some cases. It's something you could see or record and measure. Phenotypes are encoded for by the genotype. So a phenotype is the result of one or more alleles. Natural selection works on the phenotype, and if a phenotype is successful, then the alleles that code for that phenotype are going to become more frequent in subsequent generations. The Hardy-Weinberg equation describes a non-evolving population. If the HW, the Hardy-Weinberg conditions, are not met, which they usually aren't in natural populations, then microevolution may result. This next concept is so important, I've put it in yellow. Alleles which encode traits that make reproductive success more likely are themselves more likely to be preserved through each generation. But luck, good or bad, may also play a big role. So this here just kind of sums up what we've been talking about. Heterozygotes are important because they preserve alleles, forms of genes, which encode traits which are deleterious perhaps, that means they're harmful, or maybe they're just not useful at the moment. These traits may become beneficial at a later time. And this is one way that variability is maintained within a population. And finally, our terminology. When you're studying for any one of my tests, this is what you should focus on. Start here and make sure that you understand these terms and you're able to define them and compare and contrast them. Okay, that's it for today. And I'll see you again when we talk about speciation and macroevolution. Cheers.